Okay. So good afternoon, everyone. Good morning, good evening, wherever you're joining from. I'm Christiana Dapliga, and I'm a Ghanaian. I'm a trained microbiologist. I had my first degree in microbiology, and also my master's degree. I studied Salmonella, Typhoida Salmonella, in clinical microbiology from Kwame Nkrumah University of Science and Technology. And I'm currently pursuing my PhD. And I'm interested in phages. So I've been working more on E. coli and Salmonella phages. But before we start the talk for today, since I'm a Ghanaian, I will just want to introduce you to Ghana, just to see a little bit or to know a little bit about Ghana. Ghana is a country located in West Africa, a small country of about 30 million people in population, and we share borders with Burkina Faso, Togo, and Cote d'Ivoire. We have about 16 regions, and I'm talking to you today from Kwame Nkrumah University of Science and Technology. This was the university that hosted the first West African phage workshop, and it was hosted in my Department of Theoretical and Applied Biology. Also, talking about Ghana, we have quite a lot of tourist sites you may want to visit whenever you visit Africa or West Africa. We have the Boti Falls in Eastern region. We have the Lake Volta for those who like water. We have the Elmina Castle in Cape Coast for those interested in history and want to know, if you want to know more about slavery, there you go at Cape Coast. We are at Mole National Park in the Savannah region. Also, where I am in Ashanti region, we have the sword site, the Konfuanochi sword site, where the late Muhammad Ali visited and tried to pull out the sword. It stuck to the ground and he couldn't even get it out. So maybe when you visit, you can come around and see if you'll be able to pull out the sword. And we in Ghana, we also pride ourselves in our culture. So we have this cloth called kente. This one, the multicolored one, looking so beautiful. Whenever you see anything in Africa, it's usually printed on it. You can get yourself one. We have the wooden. We also have the smoke. For the smoke is the last one behind with the stripes down there. That was what the late President J.J. Rawlings loved to wear to represent Africa. And also in Ghana, we like, oh, I can't see the last slide. Oh, one more slide. Excuse me, something just. Okay, I was just going to talk about the last slide about our food. If you're familiar with West Africa, you know that Nigeria and Ghana has been on the jollof war for some time. So we have this delicacy called jollof rice. So whenever you visit Ghana, you can try taking our jollof rice. Having said so much about Ghana, I would like to introduce to us our guest speaker for today, Dr. Ivan. Dr. Ivan is the founder and the chief executive officer of Phage Genomics and one of the inventors of Phage Genomics. One of their core technology is mainly applying proximity ligation data to different facets of genomics. He received his Bachelor of Science degree at Brandeis University in 2001 and his PhD at Cornell in 2007, and went on to do his postdoctoral work in the Department of Genome Science at the University of Washington in Seattle. He has authored more than 40 academic papers and a number of patents in the field of genomics and synthetic biology. He founded Phage Genomics in 2015 and leads the organization in an executive as well as technical role. So Dr. Ivan is in and is ready to talk to us. Without much, without further delay, I think Dr. Ivan, you have the floor. Thank you.
Thank you so much, Christy. Thank you guys so much for inviting me to give this talk. I was just saying in the beginning to Christy and Jessica how fun it is sort of in these post-apocalyptic days to give talks all over the world without having to actually travel. And so let me share my screen. I'll get into it. As Christy said, my name is Ivan Liachko. I'm one of the founders of Phage Genomes. We're a small company in Seattle, in the state of Washington, in the United States. And essentially, what we do is we develop neat new genome sequencing tools for a variety of applications. And what I'll talk about today will be a set of tools we've developed that is sort of useful for a lot of different things, but specifically focus on the unique things we can do in the space of microbiome and phage discovery. So I don't know how many of you are familiar with genomics. It sounds like this is a relatively knowledgeable genomics crowd. So I just have a very small, very brief introductions to the genomics space. It's summarized in this image. Of course, what we want to do is we want to sequence everything, and we want to get as much genomic information as we can in the world for a variety of applications. The problem is this is harder than it looks, especially in the metagenomic space. You know, a lot of work we do is with single genomes, plants, animals, that sort of thing. But in the microbiome space, the main challenge is that because you're simultaneously sequencing many organisms at once, it's very difficult to when you get a mixed sample and sequence it, it becomes a challenge to try to understand where all these sequence pieces belong, right? When you shear DNA from lots of organisms at once, you have this mixed alphabet soup of shotgun sequence contigs, and then trying to reassign them to where they're coming from is difficult. There are methods for doing this called binning, but you know, if you're working on organisms that have more than one chromosome, or if you're working on mobile elements like plasmids or phages, it's very difficult to try to figure out where they are, because when you sequence this mixed community, you know, if one of these contigs is a phage, how will you know that it came from the green cell or the blue cell, right? Or maybe the green cell and the blue cell. If it doesn't integrate into the genome, it's not part of the genome. It's very difficult to track mobile elements in complex microbial communities. And so in the end, what ends up happening is usually, you know, if you take a sample, you sequence it, a lot of your reads will basically have to be thrown out because you don't know what to do with them if they don't hit something that's already in a database. So, and so what we want to do is, you know, try to make a method that can deconvolve metagenomes without relying on reference databases, without relying on a priori sort of indirect metrics like, you know, tetranucleotide frequencies and co-abundance numbers, things like that. It would be great if this method worked on phages, eukaryotes, prokaryotes, everything at the same time. It would connect mobile elements with their hosts. That's the key here. It would be nice if it did so without having to culture or cell sort or do any of these super sophisticated things. Didn't need a special machine for you to buy, and you didn't have to kind of treat it with kid gloves, like trying to extract high molecular weight DNA for nanopore or PacBio. You know, a lot of times you can't freeze the samples, all this kind of stuff. And so the method I'll tell you about today checks these boxes. And in order to explain sort of what the method is, I have to back up a little bit and go into sort of where this technology comes from. So this the method that I'll be talking about today comes from the following sort of observations. So, you know, your chromosomes, this is how you kind of, if you Google chromosome, you get something like this, but that's not actually what your chromosomes look like. They look more like this. They look like this three-dimensional structure, this three-dimensional like ball of spaghetti. And within this three-dimensional structure, there are a lot of really useful and important biological features. There are epigenetic regulation modules. There are topologically associated domains, AB compartments, enhancers, promoters touching each other. All that kind of stuff is happening. And so a number of tools were developed to understand the three-dimensional structure of a genome. These tools are generally called there's some form of proximity ligation tools. The most common of them is Hi-C. And so I'll be saying the word Hi-C a lot. I'll be saying proximity ligation a lot. In this context, it's kind of the same thing. The way these methods work is you take your cells when they're still intact in some sort of a three-dimensional structure, and you treat them with a chemical that's a cross-linker. So usually this is formaldehyde. And what it does is it goes inside the cells and chemically cross-links proteins, DNA, RNA, all the stuff that's kind of together. And in effect, what it does is it covalently binds sequences that are close to each other in the three-dimensional space. So you have two sequences, red and blue, and they get kind of glued together. We then fragment this cross-linked chromatin with usually it's restriction enzymes or DNase, some sort of endonuclease, exposes these ends. Then what happens is you do this thing called proximity ligation. So the concept behind proximity ligation is simply that if you have two DNA molecules that are physically close to each other, they're going to ligate. They're much more likely to find each other, and they're much more likely to ligate together than to some other sequence that's somewhere else. And so, because you've constrained DNA molecules close together, when you do a ligation, these pieces that are next to each other will stick together. And when they do that, so part of the protocol is biotinylating these ends, you now have this chimeric junction between two sequences, and there's a biotin in the middle. And so you can use that to fish out these junctions. And so when you fish them out, you purify them and you can sequence them with luminous sequencing. And so what this does is it allows you to say, okay, what is this red sequence? What is this blue sequence? They're in the same molecule. That means they got ligated together, which means they were physically close together in three-dimensional space. Okay, so you're saying, what is the sort of a sequence red and sequence blue? You know that they were physically close to each other in 3 dimensions before the cells were broken. And so, because you're doing this with Illumina, you're doing this millions and millions of times, you're simultaneously sort of measuring how often every sequence is touching every other sequence. So you're basically saying sequence A and sequence B were always close together. Sequence B and sequence C were never close together. And you have that for all combinations of all coordinates across the genome. Right, you're measuring how often every part of the genome is close to every other part of the genome. And then by doing that, once you've counted that, you can put these matrices together, and and then you can reconstruct 3D models of the genome. And that was sort of the original application of the technology. It's still used for this. There's a very robust field, studying three-dimensional architecture, of genomes. But there's more you can do with this. So there are two other things. There are two sort of non-standard ways of using Hi-C data that was not sort of part of the three-dimensional architecture story. Basically, Hi-C data tells you two things in addition to the 3D stuff. It tells you which sequences are close and far from each other in the genome, on the chromosome. And the reason is that if you have some kind of linear molecule, let's say imagine you have a piece of spaghetti and it's floating around in space. If two points are close together on that spaghetti, they're going to touch more often. They're going to more likely to occupy the same space. And if you're on opposite ends of that molecule, you're less likely to be occupying the same space. Another way of saying that is, if two sequences are close in 3 dimensions, you know that they're also close on 2 dimensions. So they're much more likely to be on the same chromosome, and they're much more likely to be closer together on that chromosome. So it's akin to a genetic map, right? It's sort of like if you're far, you touch less. If you're close, you touch more. And you know that for the entire chromosome. And this allows us to do things like scaffold chromosomes, phase chromosomes, detect structural rearrangements in genomes, that sort of thing. And more specifically for the microbiome application, this technology gives you the ability to tell which sequences are starting inside the same cells. Because what you're doing is you're cross-linking your sample before the cells are broken. So anytime sequences interact like this, they must have been inside the same cell in the first place, in the beginning. And that's sort of, I will expand on that in the next few slides. So just briefly to brush up against the first sort of application of the technology, if you know which sequences of your genome are closer or further together, it allows you the ability to reconstruct the genome from pieces, right? So if you're sequencing, let's say you're sequencing a genome of a plant that nobody's ever sequenced before, right? Then, can you guys hear me? Somebody said in the chat that
I can hear you.
Great. Okay. So if you have a, let's say you have a plant that nobody has ever sequenced before, and you go, you get some PacBio reads, nanopore reads, etc. You have contigs, and you want to put them into chromosomes. If you have Hi-C data, you will know which pieces belong on the same chromosome because they will be interacting a lot more. And you will know how to order and orient them because you basically arrange them in such a way as the closer the sequences are, the more they interact. So you arrange them in such a way as you're maximizing your signal. So the things that are touching more should be next to each other. Things that are touching less should be far from each other. And that allows you the ability to order and orient all the contigs along a chromosome. And it allows you to scaffold the genome of pretty much any size. Yeah, this is the visual kind of representation of that. You take, if Hi-C maps are usually these matrices that if you start with unscaffolded contigs, they kind of are a mess like this, and then you try to get computers to rearrange your data in such a way as to recreate this nice Hi-C map with this robust diagonal. The diagonal means self interactions. So the X and the Y coordinates are, the X and the Y axis is the same, it's just positioned across chromosomes. And so when you have a nice strong diagonal, that means sequences that are close together are touching more. Okay. And so this is something that I won't go into depth in. This is something we've done for years. It's kind of how we started the company. And basically we've done it on everything from humans and goats to plants to, platypuses and things like that. So for the most part of the talk, I will talk about the main part of the talk. I'll talk about the application of this technology in metagenomics with the focus on phages. So the main takeaway from this whole talk, this may be the most important slide. So I'll kind of walk through it slowly. The principle of the technology is that you're sequencing these junctions, these red, blue, right, Hi-C chimeric junctions. When you sequence one of these junctions, it tells you that whatever this red sequence is, whatever this blue sequence is, they were close to each other. But it tells you something else. It tells you that whatever the red sequence and whatever the blue sequence is, because the crosslinks happened before you broke the cells, remember the crosslinks are happening before the cells are lysed. And because of that, if you have two sequences on the same chimeric Hi-C junction, they must have started inside the same cell because they were stuck together before the DNA was released and mixed up, okay? And that's the principle behind what I'm going to talk about. And so if you have a metagenome, like you have a mixed community like this, and you shotgun sequence it, then what's going to happen is, right, so you have these contigs and you don't know where they're supposed to go. But if you have proximity ligation data, what it will do is it will create physical junctions between sequences inside each chromosome and between chromosomes as well. Okay, so you're going to get intra as well as interchromosomal Hi-C junctions, and then you'll be able to know which contigs are touching each other, and you can separate them. You essentially bin them into genomes. Okay. This is the first ever pilot project we ever did. This is a proof of concept. We published it a long time ago. Basically, we took a bunch of yeasts, 12 yeast species, mixed them together. Remember, eukaryotes are very difficult to bin in the metagenomic way because they all have multiple chromosomes and their genomes are much bigger and things like that. And so 12 yeasts, we do shotgun sequencing and then Hi-C. And so basically every one of these dots, every one of the nodes in here represent a contig. And so if you didn't have Hi-C signal, this would just be a gray field of disconnected dots. But the edges, the lines connecting the dots, represent Hi-C signal. So you can see the Hi-C signal grabs these dots, right, these contigs, and it pulls them apart into these tight clusters. Right? And when we say, well, what are the clusters? Well, it turns out the clusters are, in fact, the 12 genomes that we put in here. And so every cluster is a group of contigs that corresponds to genome A, genome B, genome C, etc. And so we showed even early on that you can do something with this technology you just can't do with traditional methods. But there's actually more you can do if you're so inclined. When you do Hi-C on a mixed community of organisms, you're getting Hi-C data for all the members of that community at the same time. And so all these purple contigs, you can say, are part of this purple genome. But you have Hi-C data for the purple genome. So you can actually then zoom into it and actually use that same data to reconstruct the diagonals on this heat map. So you can essentially scaffold this genome into full chromosomes right out of a metagenomic sample without having to have any a priori information. So you can separate genomes apart and you can scaffold them into chromosomes, and you can do other things. You can map centromeres and fungi. You can do 3D modeling, et cetera. Everything you can do with Hi-C data. And so this is a very powerful tech that, we started collaborating with lots of people. We saw what it could do. And so we went on to commercialize it to make it easier and cheaper. People, give people the ability to actually access this technology without training their entire lab and things like that. And so we commercialized this under the moniker of Proximeta, proximity-guided metagenome assembly. And it works essentially how you see here, right? You cross-link a sample, you generate a Hi-C library, and then you use the data to deconvolve a metagenome. So what I'm going to do now is I'm going to show you the sorts of things we can do with it and sort of how the platform works and some published data. So first of all, the outcome of having this kind of data set basically allows you to outperform like every binning method out there. So Proximeta is our method. What you're looking at here, this is for a particular sample, the genomes, the MAGs, right? Metagenomically assembled genomes coming out of a given sample. And basically, this figure is just showing that in terms of the numbers of genomes and the completeness of genomes, Proximeta smokes all the other assemblers out there. This is not surprising because the way Proximeta is built, it essentially takes into account normal binning information and Hi-C information and sort of puts together the best of both worlds solutions. So it sort of leverages all the development that's been done on the metagenomic binning side, and then it juices it. Basically, it supercharges it with Hi-C data. And so we get lots of more genomes separated apart. We've shown that they're more accurate than the regular MAGs. This figure basically shows the same thing, that there are orange dots. This is from a particular sample. Orange dots are genomes that only come out with Proximeta and not with traditional binning by things like MetaBat. So we get more genomes. Here's sort of a visual representation. This is how our platform looks. So we started doing this by sort of plotting traditional things, and then we integrated everything into an automated platform on the website. And so basically, the way to read this is one sample. It's a fecal sample, and what you're looking at is on the x-axis, every tick mark represents one of the genomes, one of the MAGs that came out from this sample. And the blue dots represent completeness according to CheckM, and orange dots represent contamination or marker gene overrepresentation according to CheckM. And the gray bars are a metric of novelty. So what you're seeing, so like in this sample, for instance, there are 60 genomes, 60 MAGs that are over 95% complete according to CheckM. And again, this is done on a single sample. There's no dereplication. This is just sort of it's in isolation. The sample is treated in isolation. And the other nice thing that I like to show people is, so if you have something that has a gray bar like this, what adds up, that's a novelty metric. And so basically what it's saying is any one of these bugs that have a gray bar are not matching anything in RefSeq. And so, you can see like this technology is really good at getting into that sort of quote unquote dark matter of the microbiome. There's other interesting observations, like if you go into this tail where the completeness goes down. There are actually a lot of genomes in here that are very complete, but because they're very sort of novel, Checkm can't assign them a completeness score, has no idea what they are. So remember, this method will get you not just prokaryotes, it will also get you eukaryotes, it'll get you algae, it'll get you protists, it'll get you diatoms. It doesn't know what you're assembling. It just says sequence A and sequence B were touching each other, put them together into the same bin. Let's see, somebody raised a hand. I will address the phage question in chat at the end. I think a bunch of my slides later will answer your question. Okay, so this is the so this is what the platform looks like. Let me show you another example. Some people, there's been more and more work on long read metagenomics, and if you are fortunate enough to be able to generate a lot of long read data, it works really well. Here's what it looks like when you have a high covered PacBio hi-fi assembly combined with Proximeta. This is a sheep fecal sample that we published with the USDA, with a group of the USDA. It was assembled with long reads. And so the first thing you will notice is how many mags are coming out of this one sample. So there's almost 1,200 mags that are in the sample. A lot of them are actually phased strains of the same organism and things like that. But furthermore, there are 400 mags in this result set that are over 90% complete. So one sample, 400 mags, 90% complete. And the other thing that my personal favorite is that, remember I mentioned the gray bars are novelty. You notice when you're working with a human sample, you get a lot of genomes that are known, but as soon as you go anywhere else, like you go into the sheep sample, almost everything has a gray bar, right? Meaning almost every species in this sample has not been previously sequenced. And so it's just a great way to generate tons and tons of novel genomic information. So, but we can go further than this, right? Remember, we're talking about mobile elements, we're talking about phages, we're talking about plasmids. And one of the reasons, for example, antibiotic resistance is so hard to follow is because it travels by horizontal gene transfer, it travels by plasmids, it travels by viruses, and other more sort of conventional horizontal gene transfer methods. And if you sequence, again, if you're sequencing a complex community, you can't really tell where plasmids reside because unless they integrate in the genome, they're free-flowing molecules. And the same thing with lytic phages, right? If they're floating around, you don't necessarily know who the hosts are of these phages unless you're going to culture them and isolate them and do a lot of testing sort of manually. And so what we can do is, in our sort of with our method is we can take an assembly and not just bin it like the normal way. We can actually annotate antibiotic resistance genes, plasmids, and then figure out who are they touching. So if we annotate a bunch of plasmids, we can say which genomes are these plasmids interacting with? Which genomes are the AMR genes interacting with? And sometimes we can even say, can we tell if an AMR gene is being carried by a particular plasmid or phage? So this is an example of a sewage sample. This is a wastewater sample we published with a group in Idaho. And basically the data looks something like this. So what you will notice is this circle, there's a number of different ways of sort of showing how the results look. Basically in the middle here in the circle, these are the mags, the genomes that we pulled out from the sample. And then on these concentric rings, you have plasmids, integrons, and ARGs, antibiotic resistance genes. And so if you have a tick mark like this, that means that this antibiotic resistance gene is present inside of this bacterial genome, is touching that genome, okay? So if you then have, a row of them like this, that means that this antibiotic resistance gene is present in all these different hosts. Remember, it doesn't have to be one, right? These are mobile elements that are transmitted by HDT. So they can be present in multiple hosts. And that's what's neat about this tech. It doesn't require a sequence to be inside of one organism. They can be touching, you can really figure out the host range of a given mobile element. And then of course, you can look at it this way too. Like if you read it this way with this line, this tells you that all these different plasmids are coming out of from the same bacteria, whatever this bacterium is over here. So this is potentially a way to identify multi-drug resistance, et cetera, without having to culture anything directly in a super complex community with 1,000 bacteria in it. And there's many other ways to show it. So the previous slide I showed was it was just proteobacteria, but you can do the same thing with bacteroidetes, firmicutes. You can separate these things however you want. And again, you can look at the genomes and which antibiotic resistance genes are touching. So there'll be like a cluster of them. Let's say this clade has, all these AMR genes. But we can actually go further than that. So very recently, we added this feature to our platform that allows us to simultaneously not just bin the genomes, right? Separate the bacterial genomes, affiliate the AMR genes to their hosts, but also we can say if that AMR gene is being carried by a plasmid or it's integrated in the genome, or once in a while they even look like they're being carried by a phage. And so we can sort of simultaneously deconvolve the bacteria, deconvolve the AMR genes, assign them to host, and figure out how they're being transferred. And then we can do the same thing with phages. So in 2017, Roman Kazul's lab in Paris showed that you can use the same idea to affiliate not just plasmid, but actually phages. So here you have phages on the x-axis and then bacteria on the y-axis. So you can see which phage sequences are interacting with which hosts. And I believe this is a mouse sample. And we can apply similar logic to this is the same wastewater sample I showed with a big circle, only this time we're looking at viruses, phages, instead of AMR genes. Okay, so and then this is very recent. So this was published just a few days ago. This is a paper from Tang Zhang's lab in Hong Kong, and what they were doing is they were using different kinds of methods to try to basically affiliate, try to get a method for affiliating phages and AMR genes in complex communities in wastewater. And what they did was they used, our platform, and they also used these CRISPR tools where you use annotation of CRISPR elements to try to figure out which viruses infect which hosts. And just a couple of I could just kind of highlight this stuff without going too much in depth, but basically what you will notice is that when there's a virus host sort of pair that is present in both the Hi-C data and the CRISPR data, you look to see, and they're over 90% consistent with each other. But one of the things that I want to highlight is just how they differ in the number of things you can get, right? So in an Illumina metagenome, we get 4,000 viral contigs, 1,700 of them get placed to 200 bacteria, and so whereas the CRISPR method will pull out 20, and so this Hi-C method, because it gives you this direct physical contact information, is able to recover things that on the sort of the precision front look very similar to what you would get with CRISPR predictions, but you just get a lot more of them. And so that's sort of the story behind this paper. And then we decided we could go one step further. All right, I'm going to check the chat. There's some stuff coming up. I will answer the chat questions afterwards. So in the previous slides, I shown you that we can take a phage that we annotate and figure out where is it coming from, which organism it's touching. But what we figured out is that there's more you can do. So the idea is that if you have a metagenomic community with a phage infecting some bacteria, if you have two sequence contigs that are if you have two sequence contigs that are in the same phage, they will be touching each other, right? But they will also be touching the same host, right? They touch each other and they touch the same host. And if you take both of those pieces of information together, it allows you to not just connect the phages to their hosts, but actually de novo bin phages like you would mags. And this is something that's very difficult to do without this kind of information. And so what we did was we built a tool called Proxy Phage. It's now integrated into our platform. And what it does essentially is a combination of of the sort of normal metagenomic assembly and Hi-C-based metagenomic assembly that simultaneously does metagenomic binning of bacteria and viruses. And essentially what it does is it bins bacteria, viruses, measures the interactions between them, and uses those to also bin the phages. And so you get not just bacterial mags, but you're also getting viral mags at the same time. And so here's a shot, just a figure to kind of maybe drive it home a little bit. What you're looking at here, this is from a fecal sample. Every pie slice in this chart is a VMAG, is a viral mag, okay? And so within this pie slice, there is this line. And what that line is, these are a bunch of contigs that are touching each other, but they're also touching the same microbial host. That's what this line means. And this line here, that means these contigs are interacting with each other, but also with the same host. And so they're coming, these little lines, they're basically viral genomes, viral mags, okay? And so we're able to sort of pull viral mags directly out of a metagenomic sample. But more than that, we're able to connect them, because remember, we're simultaneously affiliating them with the bacterial hosts as part of the process. And so we get the same thing where here, these phages on the top and bacteria on the vertical axis. And so you get these like little bleeps on the heat map. That means that this virus is interacting with that bacteria. Okay, and so you can tell immediately the host range of all your phages. So you're simultaneously deconvolving these bacteria genomes. In many cases, they're novel. You are binning the phages, which almost always are novel, and you're simultaneously connecting them together, which phages come out of which bacteria. And you get cool things like co-infection, where you can detect, if multiple phages infect the same host. You can look at phage host range, so like promiscuous phages, where you have this, the same phage can infect multiple different organisms, multiple different bacteria. So you get all this out of the same data set. And we've done a lot of benchmarking because, of course, this is a novel method. And so, how do we know that what we're binning is real? So basically what we've done is we've used things like CheckV to benchmark our viral mags, and that sort of have cutoffs in the software to kind of filter things. We've also done a lot of benchmarking against long read assemblies, right? So one beauty of a long read assembly is that your contigs are so long that oftentimes a phage will be all encompassed in one contig, an entire phage genome. And so this allows us to take our short read mags, viral mags, and map them against long contigs, long viral contigs from the same sample, and compare how often they're accurate. The numbers come out to be like 96% in agreement with long read assemblies. It looks something like this. This is another way of looking at it. So this is a fecal sample that we processed, and basically we pulled out from this fecal sample with Illumina data 73 high quality viral mags that passed all the kind of check marks and thresholds. And so we got 3 high quality viral mags from the Illumina assembly. From the same sample, we had a nanopore long read assembly, which had 25 phages. So these 73, we found 25 phages that the 73 mapped against. So 25 phages pulled out by ONT, 73 phages pulled out by Illumina Proxy Phage, and then you map them against, you kind of see this. So the triangle in the middle of these little stars is this is your ONT contig, and the circles are the Illumina contigs. And so basically the idea is these are all supposed to be kind of mapping to the same ONT contig. And predominantly they do. There are a couple of notable exceptions where it's broken, or if you have contamination, but overall, 73 mags, 25 of them map to ONT, 19 in perfect agreement with ONT data, 2 of them have contamination. A lot of times the contamination is caused by when you have more than one phage infecting the same cell. And then of course, the Hi-C data doesn't know, right? So it tries to group them all into one. So it creates these like hybrid bins like this. And so one note, this is actually a note for myself to point out another important thing. So we love long read assemblies. ONT assemblies are great. PacBio assemblies are great. However, one benefit of having short reads is that you get a lot more things. So in this case, right, so we got about 3 times more viruses from the Illumina assembly than from the ONT assembly. And we probably spent half the money, right? So you get way more stuff because Illumina reads are cheap, and they can really get down, and they're numerous, and so they can really get down and pull out things that are low abundance, much better than long reads. But of course, long reads give you the nice, long, beautiful context. And so this method applies to all sorts of things. So here, I showed you before a fecal sample. Here's, this is that sheet paper with some enormous, hundreds of phages. Here's another human fecal sample. This is cow rumen. This is wastewater / sewage. And so you can see like every one of these samples we go through, just tons of phages and their hosts. So and this is of course not mentioning all the different bacterial mags that are getting pulled out. So lots of new phages, lots of new mags. And this allows us now to do some really cool experiments. So this is a time course where day 0 is sort of the end of an antibiotic course. And so this is recovery from antibiotics over time. Fecal samples taken every few days after the stopping of an antibiotic course. And so, not surprisingly, with time, you see the new bacteria, MAGs, right? New metagenome assembled genomes are popping up. So your flora is becoming more diverse. It's sort of recovering, right? And at the same time, viruses are coming in, right? So as the bacteria populate the sample, new viruses are coming along for the ride. At the same time, AMR genes are going away and plasmids are going away. Again, this is not surprising, and this is sort of what you would expect in a normal kind of antibiotic recovery course. But remember, we have more information than this. We can actually go into all these samples and not just look at abundance of particular taxa or genomes. We can actually also ask, what's happening to the mobile elements, right? We get this, we get viruses, we can tell who the hosts are. Either as contigs or as mags. And what I didn't tell you, but that's very analogous, is the same thing that happens for viruses is also happening for plasmids. So ProxiPhage is actually also proxy plasmid. It's binning viral mags. It's also binning plasmid mags using the same kind of the same logic. And so you get plasmid host range, you get virus host range, all at the same time. In addition to getting all the mags. And so we can do things like this, right? So that day 0 through day 29 that I showed you before, you can actually look at these communities in a much deeper way, not just the abundance of particular taxa, but actually what are the phages doing inside of all those communities over time. You can see them, not just in this case, there are fewer here and more here, as we saw in those pink heat maps, but you can actually see them move around. And the same thing with plasmids. Again, we can go from right. This is the contig way of looking at it, but you're also able to look at the presence and the host range changes in the plasmids. And of course, we can bring it all together. I showed this figure before where we can see sort of what the antibiotic resistance genes are doing inside of these samples, and we can do that over time. Right, we can see, like, for example, here in the beginning of the recovery, there's lots of yellows. Those are plasmids, lots of plasmids. And then by the end of the experiment, most of the plasmids are gone. And so the antibiotic resistance content is going away, but specifically the plasmids are getting lost much faster than the integrated genes, as you would expect. Okay. So I've shown you guys a lot of information. And just to summarize it all, basically, what I set out in the beginning of the talk is to make a method that's simple, but gives you tons of useful information. And so you're getting all these genomes, all these viruses, all these plasmids without any culturing, without any a priori information. You don't need high molecular weight DNA. You don't need a special machine to buy. This is just a kit. So we sell kits for these things, and all the analytics that I've shown you are they're included for free on the platform. So everyone we work with gets all of that stuff that I just shown you with the phages and the plasmids, etc. And with that, I'm going to thank my collaborators, both on the academic and on the corporate side, and the funders. And that's it. I'll take your questions. If you guys are interested, this giant QR code is intended for you to take a picture of on your screen. And then if you're interested, and it'll take you to the publication of the phage binning and host affiliation story. And so, so that's it. And I'm happy to take questions.
Oh, thank you very much, Ivan. That was an interesting talk. As I was listening, it was interesting to find out that at the end of the talk, you don't need a special kit, and I think that makes it interesting. So my question would be, while before I ask others to come to ask the question, over costing is a problem for us here in Africa and all of that. What is the cost benefits of using these techniques for your work for a situation like ours in Africa? Thank you.
Sure. Well, so it's a very good question and a very sort of pragmatic and honest one. When we started, we were academics, right, before we started, and everybody's got some amount of cost inflexibility. Hi-C used to be super expensive. It used to cost something like $1,000 in just reagents to make one library, and that's part of why we sort of created Phage, is to make this technology easy for people to use. Why would you pay more money for something like this than doing 16S, etc.? It's because the information you get, there's sort of some limitations on the method, like we can only make it so cheap because there is lots of stuff we have to put into these kits. But I think relative to having to like there are certain things you just can't get without it, right? Like host range of phages, it's going to be very difficult to measure the amount of labor that would have to go into just processing a single sample with 100 phages in it, right? This takes you like 2 days and then some sequencing and then you're done. So I would say that the justification is if this information is of value to you, some of it you just can't get any other way. Some of it you can get other ways, but the labor would be extreme that is required.
Yes. So for others, I don't know how the question nature it will be. We don't have much questions. I don't know if you can look through the chats and respond to the questions.
Sure.
Yes. I should ask the question.
Let's see. So I'm scrolling up. There were quite a few. Okay. Let's see. So one question was, can this technology distinguish if the genomic change is by the resident phages or by the newly? So we have done experiments. Folks are asking, can you do experiments if you introduce a phage into a community and figure out who it's infecting? And the answer is you can. We've done some of these experiments and everyone is different, but you can imagine synthetic community of different bacteria. You introduce a phage or a cocktail of phages, let them infect for 15 minutes or so, and then cross-link and then do this kind of analysis. That's a totally legit thing to do. And it's relatively inexpensive because you don't need a lot of sequencing. So it's potentially another way of kind of, if you already have phage cocktails or individual phages that you're interested in measuring the host range, there are sort of simpler ways of doing it without having to annotate everything, right? If you already have all their references, it makes it much easier and cheaper. There was a question of, have we done this with toxic genes? In principle, it doesn't matter what the gene is. You can annotate any genes you want in your metagenome, and we can figure out where it's coming from. So whether it's like, Shiga toxin or some other toxicity gene that's being spread by mobile elements, you should be able to detect them. We kind of focus on phages and AMR genes just because they're such broad categories of things that people work on, but this can apply to any gene you want. In fact, when one of the things our platform automatically delivers is a full metabolic workup of all your mags, and so you're going to get all the biosynthetic pathways, everything that comes out of all these mags.
So Ivan, I'm thinking, because the question is being recorded, so is to ask questions so that people can know the questions that were asked and you're answering them. So let me just answer the question. Let me just ask the question here.
Right. You ask, I'll answer. It'll be easier.
Yeah. So we have a question from Joseph Michael asking, how sensitive or effective is the platform when scanning for transposons hidden within the Liachko Phage genomes?
So I actually don't have an answer to that one. One of the issues, so we can definitely if the transposon is unique, we'll be able to find it. The problem with is that this method is sequencing based, and so if you have a transposon that's identical, it's present in many copies, it'll be difficult to distinguish them, and so we're limited to looking at unique reads, uniquely mapping reads, and so I think I would not make too strong conclusions out of transposons because they're usually present in many copies all over the place.
Okay, do you want to follow up on your question? If you want to follow up on your question, you can unmute. Okay, we have another question from Newton saying, what are the thresholds of completeness and contamination below which the sequencing data are considered as poor quality using your Hi-C tools?
You know, it's whatever you want. Our thresholds, our completeness contamination thresholds are kind of what people use normally, you know, like 90%, 70%, but those are, you know, and contamination wise, like everything has to have below 10%, but we report everything, right? So you will see the good genomes and the bad genomes, and you see all of them, and you can decide what's high quality, low quality for you. We make sort of, you know, we make it like we put like colors on these things, but it's very arbitrary. You know, we sort of decide based on what most of the field uses.
Okay, and Stephen asking, what percentage of viral reads map to the HQ phage contigs? Is it low because you have to catch the phage infecting the bacteria?
Yeah, it's a good question. I don't know numbers offhand, but it is true that yes, right? So a phage, you know, will only spend time in the bacteria for a certain amount of time. Then it lyses it, you know, breaks open. You know, whereas plasmids, something like a plasmid, kind of live there all the time. And so we do see in many cases fewer reads hitting phages. But the nice thing about Hi-C is that a little goes a long way. So if you know, the data is kind of bimodal. It's either very, very strong or very, very weak. So if you have even a little bit of real junctions, real connectivity information between a given phage and a given host, we know that it's there. And so it's a little bit less like you know, with shotgun, if you just sparsely pick up a genome, it won't assemble, right? That you need to have a certain threshold of connectivity before you get it. That threshold is much lower with Hi-C because you're just connecting dots. And so you don't have to have kind of like tons and tons of reads mapping to the same thing. But I don't have a quantitative answer to your question, unfortunately, because it's, you know, in part because it's different for every sample, in part because I don't know.
Okay, Joseph is asking, what's the minimum DNA mass required for the work for this?
Sure. So one key concern here is that one key feature of Hi-C is that it needs to start with intact cells. So we don't measure input in terms of DNA mass like you would for a shotgun library. We measure how many cells do we need to get started. And the answer is, if you have a sample that's pretty biomass rich, like it's not like soil with a little bit of bacteria, if it's like mostly bacteria like fecal or rumen or sewage or something like that, then usually we ask for a couple of million cells. So like 10 to the 6, 10 to the 7 is fine. If you have more, it's great. Obviously in a fecal sample, you know, if you get 10 to the 7, 10 to the 8, that's great. So, but at least a few million cells.
Okay, and Stephen is saying, are there issues with cutoffs of network building? For example, a phage that infects one bacteria versus the one that infects many?
Well, we can see that, right? So we can see if a phage infects multiple or one. And with cutoffs, I mean, definitely an enormous amount of the sort of algorithmic development in the background is specifically to deal with those cutoffs. So yeah, so if you look, how can I answer this the best way? It's definitely something we've taken into account. And so there are issues, but we've dealt with the issues, right? So within the algorithm. So if you just look natively, then yes, if you just kind of make a naive case of contig A touches contig B, C, D, that's not enough. You have to normalize it in a special way. And there's lots of other sort of algorithmic things that have to be done to do it properly. But it's something that we've spent a lot of time putting together.
So I think Newton is asking a follow-up question to his question, and I just to add a little bit background to that. For plant genome, for example, we've tried to see how Hi-C is used to map completeness. So you have your full genome, you do long-range sequencing, possibly a hybrid assembly, and you get a Hi-C to phase it, not to phase your genome. Can you do phasing for phage genome? So it's Hi-C assembly, is it an assembly tool or annotation tool or for both? So can you do a phasing for a phage genome using Hi-C in combination with a Illumina high-read or hybrid sequencing?
Sure. So there's two questions there. One is Hi-C assembly a tool for annotation or assembly, and the other one is, can you phase phages? So the way to summarize what Hi-C does is you take an existing assembly and you think of it as a jigsaw puzzle with lots of pieces, and Hi-C tells you which pieces are next to each other. And so it allows you to put things together. So Hi-C is neither an assembly tool nor an annotation tool, but it can be used to do both. So once you have your genomes put together, right? So the assembly happens before Hi-C is involved. Assembly is when you take shotgun data, you PacBio reads, nanopore reads, Illumina reads, and you make the contigs. You make the jigsaw puzzle pieces. And Hi-C takes the pieces and puts them into genomes, separates them out, connects phages and hosts, etc. And once you've done that, you can also annotate them. And the annotation is done using just normal annotation methods. But once you, for example, if you have bacterial mag, when you do your annotation, you're able to reconstruct pathways. If you don't have a mag and you have lots of contigs all mixed from different organisms, you don't really know what the pathways are because you don't know which ones belong inside the same organism. So Hi-C is not an assembly tool, is not an annotation tool, but it enables both. And then as for phasing, so what the question was about is when we do like a plant or a diploid mammal or something like that, we're able to not only put chromosomes together, but also haplotype phase them. So we can actually separate the maternal mutations from the paternal mutations. The idea is that if you have chromosome 1 from the mom and chromosome 1 from the dad, they're physically two separate objects. So the unique SNPs on the maternal chromosome will touch other unique SNPs on the maternal chromosome. And the unique SNPs on the paternal chromosomes will touch other unique SNPs on the paternal chromosome. And so you can actually separate the haplotypes. And that's something we do for big genomes. The question is, can you do that for phages? Because phage genomes are very small, usually the phasing happens. So in principle, yes, you can. But in reality, what ends up happening is because phages are so small, if you have meaningful size phage contigs, they oftentimes are sort of phased before we even start. So certainly if you have any kind of long read assembly, then you would have phased them before we even involve the Hi-C, and the Hi-C just kind of pulls them into the bins. So, but we haven't really dug very deeply into that question. So my answer is theoretically, yes. Practically, it's probably going to happen automatically by itself, but we haven't gone too far into it specifically.
So Christy is asking, can Hi-C be used to determine if phage can be used for therapy? And what advantage does this method have over other methods?
Well, what this method does is it generates knowledge. It tells you which phages are infecting which bacteria, right? It doesn't necessarily make your therapy better, but if you have this information, you you could look at, you can make a cocktail that infects specific bacteria because you now know all their host range. It makes it easier for you to isolate if you have a specific bacteria you're interested in or phage you're interested in. It makes it easier for you to extract the relevant ones, right? It lets you do experiments where you can measure host range. Maybe you're trying to manufacture it and you want to make a strain that simultaneously grows every phage, right? Things like that. So that's basically what Hi-C does. It just increases your knowledge base.
Okay, thank you. And Hani is asking, does this mean that you can detect free phages in the samples? They have to be or do they have to be in the cell?
So they generally do have to be in the cell. The thing is, you can detect free-flowing phages with normal shotgun sequencing, right? So you don't necessarily need Hi-C to detect the free-flowing ones. You can just take the soup and sequence it, and you'll have your phages. We can get Hi-C data on phages that are not inside cells, but it's not clear that you would need to do that, right? You would want to, there's easier ways to get that information.
I think Anik may want to unmute to ask his question. He added something by meaning, can it be used to link? Yes.
Yeah, sorry. Wonderful approach. Amazing. I just wanted to see, I think the benefit actually for us is just to try to link the phage to their hosts. I'm talking about actually in any environment. Sorry.
You guys are breaking up.
No, I think someone actually was unmuted. So can you hear me right now?
I can, yes.
Yeah. I think it's again, it's amazing tool actually that can link the phage to their hosts, which is kind of wonderful thing. But if you are trying to track actually the lytic phages with their hosts, can that be done actually using this approach? Especially that unless if they are outside the cell and you would like to say actually that these are salmonella phages and you have salmonella and whether they are their hosts or not or something like that. Is it possible to be done?
Yeah, definitely. I mean, we do it all the time. And the idea is that you have a population and some of them are in different stages, right? Some of them are infected, some of them are lysed, some of them are integrated. And so you are capturing a snapshot in time. And so if you have lots of samples that are external, then they won't get caught, but some proportion of them will be in the cell and you're catching those.
Yeah, I think it's wonderful. I'll be in touch later on because I'm really interested actually to apply that for food samples. So it's going to be a complex matrix and all that. So I'll be in touch. Thanks.
Yep. Thank you. Sounds good. And so I see a question in the chat about finding integration sites of prophages. So we do have, so have we done this to look for prophages? So we do, that's one of the things we output. So we estimate, you can actually estimate by abundance and sort of abundance and intra-contig, mag, Hi-C signal strength, whether something is integrated in that chromosome or it's not. Because if it's not, it'll have a lot less Hi-C interaction than something that's on the same physical chromosome. And so we do, that's one of the outputs. I mean, it's an estimate. The problem is a lot of times we don't know what the truth is. And so it's hard to put numbers on it, but we do have statistics and sort of a prediction of whether or not something is a prophage or not. And then as for the sites where the prophage might be landing, it's definitely doable, but it's, the genomes are so small at this point. I don't know if the resolution might just be bigger than the genome itself. So a little bit of advanced topics. I'm happy to talk about it later if you're interested.
Yeah, I don't know if John can unmute to ask his questions. He's asking, how does this apply to organisms with large genomes and metagenomics? How does it?
Yeah, and can you clarify, John, what you mean by large genome? Do you mean like a plant with a fungal infection, or do you mean like eukaryotes, prokaryotes?
Yeah, Thank you very much, Ivan. My question was actually directed to where we are developing SNPs and of a fish called lungfish, which has a large genome, but when we try to find out, there are also some viruses in it. But because of this we're talking over 1.3 billion, base pairs, and so how feasible or flexible is this method? Can this be used?
And so what are you trying to assemble the fish genome, or?
Well, we're trying to assemble the fish genome, but also the viruses maybe that are attached to it.
Right. So assembling the fish genome, 1.3 gigabase genome is not a problem. We do fish all the time. We do big, giant, 5 gigabase plants and north of that stuff. So for scaffolding and assembling the fish genome, that's a very normal project for us. The viruses, the question, so remember, at least in this case, the viruses that I'm describing are phages, right? This is not your RNA viruses and the viruses that are infecting like a mammal or a fish or something like that. And so if you're looking for eukaryotic viruses, you may not need, you probably don't need this because, I mean, to some extent, you already know who the host is, right? It's your fish. And so you don't necessarily need Hi-C to tell you that. And there's probably easier ways of doing it, like just try to purify the viral fraction of things and long read sequence them. So I don't know I'm not sure if I'm answering your question correctly, but if you're looking at if you're looking at a eukaryotic virus that infects a eukaryote, you probably don't need this.
Oh, okay, you've just answered it. Thank you.
Okay.
Yes, I think with that, we have come to the end of the webinar. Thank you very much, Ivan. I think we can be in touch. Thank you very much everyone for participating. Jessica, Christy, thank you very much. I also know that African Phage Forum is planning for a symposium very soon. I think that will be communicated to us soon. So thank you very much for always attending and for your time. And I want to say goodbye from Nigeria. Thank you.
Thank you guys so much.
Bye, everyone.